So the first proof of concept we started with was to take healthy adults and to collect what we call mobilized apheresis product. So in this case, the stem cells typically will reside in your bone marrow, but we wanted to make this less invasive. So the standard clinical process for collecting stem cells from a person would be to give them a growth factor injection once a day for 5 days, and then on the 5th day to hook them up to an apheresis machine, which will take a unit of blood, spin it down, collect the white blood cell fraction where the stem cells are, and return the rest of the blood product back to the patient. This gets rid of a lot of the unwanted cells like red blood cells up front so that they don't influence the process. And this is also the most common blood product that is used in gene therapy trials currently. So once we had these products, we put them onto the device. Our programming takes the products initially, does the introductory preparation of these cells for cell separation, and then it actually uses a magnet to pull the stem cells out of the blood cell pool. And it does this by taking an antibody that detects CD34 that is attached to a metal bead and incubating it with the cells. Any stem cells or HSPC will pick up the antibody if they express CD34 and will also pick up the metal bead. Then we activate a magnet, which essentially makes all of those cells stick, while the rest of the cells that we don't want pass through. We then transfer the cells to the chamber highlighted on the device here. We can regulate gas, specifically 5% carbon dioxide is a mimic of the human body condition, as well as 37 degrees Celsius. And we expose the cells to the same gene transfer reagents that we're currently using in our clinical trial. In this case, it's a lentiviral vector that encodes those 3 therapeutic genes, the CCR5 knockdown gene, the C-peptide, and our chemotherapy resistance gene. We then cultured the cells and then prepared them for infusion by washing them to get rid of all of the viral reagents, the culture reagents, and then putting them into a formulation that would be safe for patient infusion. This is essentially like a saline solution that contains a pharmaceutical-grade human serum albumin. Now, we tested these to make sure that they met our criteria for infusion, but to test their functionality in a biological system, we couldn't put them back into the patients that we got them from, 1, because those patients were healthy, and 2, because we don't have FDA approval yet for the device. So as an alternative model, we actually used an immunodeficient mouse model called of xenotransplantation. These mice don't have an immune system, so these cells can be accepted and can go into the bone marrow and graft and produce blood cells, and we can track that over a period of time. To do this entire process required us to build about 5 custom software programs and to manipulate 2 different tubing sets. The tubing set is all of the clear lines that you see weaving in and out of this device in the photograph. And then to get these cells to perform in the mouse, we also had to give the mouse 2.7 gray total body irradiation to create some space for these cells to go in. But this is not fully loaded conditioning. This is what we consider mild conditioning in this particular mouse.